Okay, membranes and transport. Membranes are a fluid mosaic model, which means that the molecules within that membrane can move around each other, and the mosaic part is because of the arrangement of proteins within the phospholipid bilayer. So we have a phospholipid bilayer. The width of this membrane is approximately 7 nanometers. Under an electron microscope, that would be seen as two dark bands parallel to each other. The components include the phospholipids. You need to remember that the structure of a phospholipid is a phosphate head, which is polar, and two tails, which are fatty acids. Okay, the polar head also contains the glycerol, and the fatty acids can be saturated, so that means that they have carbon to carbon single bonds, no double bonds between the carbons in their structure, and therefore are fully saturated with hydrogen. There's no room for any more hydrogen atoms in that structure. You can sometimes have unsaturated fatty acids. They might be strewn with a kink in the fatty acid tail, and that's because there is a carbon to carbon double bond, and therefore it is possible to add hydrogen to those structures. So the phospholipid, the fatty acids are what we say hydrophobic. That means they do not like water and water-soluble molecules, and therefore repel anything that is charged or polar, such as sodium ions and potassium ions. The phosphate head is polar and therefore will interact with water, so you can have a more stable membrane because the tails stay together as this hydrophobic region in the middle and this hydrophilic region on the outside that's able to interact with water molecules. So that helps to maintain the stability of that membrane. Also within a membrane you can get cholesterol, and that helps to regulate the fluidity of the membrane. What that means is the more cholesterol you have, the less fluid the membrane is, so therefore the less things can pass through and the less movement within that membrane there is. Don't talk about it being tougher or harder. It really is. The more cholesterol there is, the less fluid it is. Okay, you can have carbohydrate attached to a phospholipid, so we call that a glycolipid. You can have carbohydrates attached to proteins, and we call those a glycoprotein. That doesn't mean, this glyco part does not stand for glycogen. Never say that it's glycogen. It is a carbohydrate attached to either a protein or a lipid. The proteins can go all the way through the membrane, and we call those intrinsic proteins. We can have proteins that just sit on the extremes of the membrane, so on the outer edges. So this is an extrinsic protein. These proteins could be enzymes. These intrinsic proteins could be channel proteins, and so help with facilitated diffusion. And as we've already said, that's because the phospholipid bilayer is hydrophobic in this region, which means that any polar molecules or charged ions, for example sodium ions, chloride ions, cannot easily pass through this phospholipid bilayer. So what they do is they can move through the specific protein, but it has to be a protein that is able to allow that particular ion through. So proteins that allow sodium through will not allow chloride ions through. Okay, you can also have carrier proteins, and they are for active transport and facilitated diffusion again. And in that case, if it is for active transport, then what will happen is you will have an ATP molecule that is able to bind at this point. As it hydrolyzes, that causes a shape change in the protein, a 3D conformational shape change. That allows then the molecule that's on this side to be moved in, and it doesn't have to therefore be down a concentration gradient, so you can actually accumulate ions or molecules within the cell, because this protein, because it needs ATP to bind on this side, will only allow transport in this direction. That's in contrast to another protein, for example a channel protein. If we say that this protein over here is acting as a channel protein, then you can have facilitated diffusion through this channel protein. If there are more molecules outside here and fewer here, they will follow the concentration gradient into the cell. However, if it gets to the point where you've got more molecules inside the cell and fewer molecules outside the cell, then you will get facilitated diffusion out. And that's because this is a channel protein, and facilitated diffusion follows the concentration gradient. So wherever the concentration gradient is, that will be the direction of flow of those molecules through that particular protein. Note that proteins can have different 3D shapes. So when you see a differently shaped molecule in the membrane structure, you will probably be best to guess that it's a protein. This one is a glycoprotein because it has carbohydrate attached to part of it. This could act as a cell signalling molecule. It could identify the cell as self. It could be a receptor molecule, and so allow things to bind to it, and then on the other side have molecules that it triggers to cause a response within the cell. So it is able to respond. For example, hormones may bind up here, and then they activate a second messenger inside the cell to cause an event to happen inside the cell. So our membrane then is made up of a phospholipid bilayer that contains hydrophobic fatty acid tails that prevent or repel the movement of charged molecules through the phospholipid bilayer. You have proteins that have different shapes. Proteins that go all the way through the membrane are called intrinsic proteins. Proteins that sit on either outside or inside layers of the phospholipid bilayer are called extrinsic proteins. These can be acting as enzymes or, again, signaling molecules and identifying the cell as self. You have cholesterol, which acts to regulate the fluidity of the membrane. The more cholesterol there is, the less fluid the membrane is. You have glycolipids, which can interact with water. They can also act as cell signalling. You've also got another function of the membrane, and that is to allow cells to be joined together to form tissues. And they do that by glycoproteins. They can join to each other and make cells into tissues, which then function. For example, you have endothelial cells which all join together to form the endothelium, which is a tissue acting to carry out a particular function. And that's membranes and transport.